Hello, and thank you all for joining us today for our overview and introduction to using real-time PCR in the classroom. I'm Ingrid Miller, Product Manager for Bio-Rad's Biotechnology Explorer program. I'd like to introduce David Palmer, our real-time PCR expert and presenter today. Dave studied at the University of Guelph in Ontario, Canada, where he received his Bachelor of Science in Agriculture. After receiving his doctorate in molecular plant pathology from Oklahoma State University, Dave performed field and greenhouse research on biotech crops for Zeneca Ag Products. He then co-founded a biotech startup company called Biotics. Currently, Dave is a senior technical support consultant for Bio-Rad Laboratories, supporting educational electrophoresis, gene transfer, and PCR products. Dave is very passionate about education and teaches an annual PCR course as an adjunct professor at Contra Costa Community College. Welcome, Dave.
Thanks, Ingrid. All right, and thanks everyone for attending this real-time PCR presentation that we've got today. Is the audio level okay? If the participants can click on the yes checkbox if you can hear me okay? All right, good. Okay, so we'll continue on then. Let me just clear those checkboxes. So today what we'll talk about is real-time PCR and a couple of different things. First, we'll go over what is real-time PCR used for, how does real-time PCR work, what chemicals and instruments are used to detect DNA, what does real-time PCR data look like, how can we demonstrate real-time PCR in the classroom, and what are some ways to troubleshoot real-time PCR. So just a little bit of housekeeping to start off with. If you have questions about the presentation, we'll try and answer them at the end of this. So questions you can send by text chat to the presenters. And at the top of your screen, you'll see a little text chat box, and you can just open that up anytime you'd like and send some questions in if there's anything you want to know about. Whatever we don't get answered live at the end of this presentation, I'll try and answer by email, and that will be attached to a mail that will be sent out after the presentation. So to start off with, what is real-time PCR and what is it used for? Well, many people here have taught PCR before, and we remember that PCR is the polymerase chain reaction. It's a process for the amplification of specific fragments of DNA. So amplification and specific are the two key components there. Real-time PCR is a specialized technique. It's part of regular PCR that allows a PCR reaction to be visualized in real time as the reaction progresses. So as we'll see, real-time PCR allows us to measure minute amounts of DNA sequences in a sample. And the basic summary of this whole presentation is right here. Conventional PCR tells us what, real-time PCR tells us how much. So some examples of things that real-time PCR is used for are gene expression analysis for cancer and drug research, for example, disease diagnosis and management, food testing. It's used in animal and plant breeding, and also forensics, and I'll go into a bit more detail on some of these. One example for the use of real-time PCR in gene expression analysis or basic research would be research into the BRCA1 gene. That's the gene that's involved in breast cancer, and that gene is involved in tumor suppression. It modulates the expression of other genes. So the way the BRCA1 protein works is it affects the conversion or the transcription of DNA to messenger RNA, and that ultimately affects the amount of a particular gene product protein that's produced. Real-time PCR will tell us how much of that messenger RNA is produced from a particular gene. So when researchers do gene expression studies and say that a certain treatment or a certain disease state upregulates or downregulates a gene, a lot of time they're getting that data from real-time PCR because it tells them how much DNA or messenger RNA there was. Another use for real-time PCR is in disease management, and an example for that would be typical HIV treatment. And in that example, what the physician would do would be to monitor the viral load or the amount of virus in a patient. And real-time PCR allows them to do that by looking directly for the amount of virus RNA in the blood. Real-time PCR will amplify the RNA and quantify it, and you'll be able to adjust prescriptions and disease treatment based on the amount of virus as determined by real-time PCR. And another example, kind of related to my background with work with the Zeneca, is in determining the percentage of GMO food. With genetically modified foods, those foods that have a foreign gene inserted into them, a lot of countries have regulations as to what the maximum amount of that type of food allowed is. And the way that you can determine how much GMO content or transgenic content is in a certain shipment of grain, for example, is to sample that shipment, measure the amount of regular or wild type DNA. I have in this slide WT-DNA for the regular wild type DNA. Also measure the amount of genetically modified or foreign DNA. And if you take that ratio, you can determine the percentage of GMO food. And that's the commonly way used to measure how much of a shipment contains genetically modified food or not. Yet another example of real-time PCR that's just kind of coming into play is real-time PCR in forensic analysis. There are some real-time techniques that can be used to identify stains. So for example, a stain on a carpet, rather than using a color change test like you'd see on the TV shows where they'd put a Q-tip on the stain and then add a reagent and see what color it changed to determine what that stain was made of. Real-time PCR can actually help determine what the composition of that stain is, whether it's human blood or animal blood, for example. Another factor that relates to forensic analysis would be how much DNA is in a particular sample. Because when DNA is subjected to genotyping, the genetic fingerprinting, a certain minimum amount of DNA is needed, and real-time PCR can help to determine whether there's enough there and how much is there. So those are some examples of real-time PCR. Now we'll go on to how does real-time PCR work? And in order to understand real-time PCR, we'll just review how regular PCR works. And quite a few of you have already taught PCR in your classes. Regular PCR, you start with the double-stranded DNA template. You also have in your PCR reaction or your PCR tube the primers and nucleotides and the DNA polymerase, along with buffer and water and all the components of the PCR reaction. That reaction tube is then heated in a thermal cycler, and when it's heated, the DNA denatures. Once that reaction is cooled so that the primers can anneal, the primers will bind to their specific recognition sites, and then the Taq polymerase that's also in that reaction will go and extend those primers with the nucleotides and make copies of the original templates. So when we start with one original template, by the first reaction we end up with 2. After 2 repeats of that cycle, we'll have 4 copies of our original template. And then after 3 cycles, we'll have 8 copies of our original template. So PCR goes on in an exponential manner, and I know many of you have taught PCR already, and you're familiar with that. When I teach PCR at the local community college, I teach over at Contra Costa Community College, I often use a nice little animation that Bio-Rad has. It's got a lot of detail on it. It's very well drawn and very, very clear, and also scientifically accurate, which is kind of nice. So it's my favorite animation. There's also animations available on the internet, like one from Cold Spring Harbor I also use that has some good information on it. And there's also a new PCR poster that Bio-Rad has come out with. In one poster has from beginning to end how PCR works, and I'll be using that next spring when I teach. I think that's a really good resource to have. So that's how traditional PCR is usually presented. And in order to understand how real-time PCR works, what we'll do today is use a thought experiment. And this is kind of a different way to teach real-time PCR than the way I learned it. I had seen presentations before on real-time PCR, and they're really hard to follow, and they didn't really make sense for how you would relate the information coming off the computer screen to quantity. So what I started doing after I had bad luck teaching that to my own class was I switched to this method, and this seems to work really well. So think about this. If you're teaching real-time PCR for your own class, you may want to try this method. So this is what we do. To understand real-time PCR, what we'll do is we'll imagine ourselves in a PCR reaction tube at cycle number 25. So we're there in the PCR tube. What's in that PCR tube? Well, in that tube, what we've got are nucleotides, we've got primers, we've got our original template that we're trying to detect. We have amplicons, the amplified DNA product. We've got our enzyme, and we've got the buffer and the water and other things in there. And just for the sake of argument then, let's say we've got 1 million copies of the amplicon right now. So we're at cycle 25, and we've amplified, and we're now up to 1 million copies. So continuing in this thought experiment, what was it like in the last cycle, at cycle 24? Well, the conditions were almost the same as what we are now at 25, but we only had 500,000 copies of the amplicon. Makes sense, right? Because PCR doubles every reaction. So if we go back to the last cycle, we only had half as much. What about cycle 23? Well, cycle 23, we probably only had 250,000 copies. And then cycle 22 is likely the same reaction in the tube, but only 125,000 copies of the amplicon. So what we can do is we can plot that on a graph. And if we plot the cycle number on the x-axis versus the quantity of DNA on the y-axis, put our first point right there. Cycle 25, we had 1 million copies. Cycle 24, 500,000. 23, 125,000, and so on. We get this nice exponential curve. And that's exactly what you'd expect from PCR. We got to that working backwards, but you can also work to it forwards, where you're amplifying and amplifying. You can't see it on the graph because of the scale on the graph, and then all of a sudden you see the curve shooting up. So now continuing this thought experiment, what's going to happen after the next cycle, cycle 26? Well, if cycle 25 we had 1 million copies, cycle 26 we'll probably have 2 million. And what about cycle 27? Well, if we had 1 million at 25, 2 million will have, cycle 26 will have 2 million. Cycle 27, we may have 4 million copies. And you can probably see where I'm going with this. What's going to happen at cycle 200? In theory, if we kept doubling, there'd be one with that many zeros. I think that's around 69 zeros, numbers of amplicons. That works out to 10 to the power of 35 tons of DNA. And that much DNA would be the equivalent in weight to 10 billion planets the size of Earth. So that's a huge amount of DNA, and obviously that's not going to happen. So have this note here, a clump of DNA the size of 10 billion planets won't quite fit in our PCR tube anymore. There's no way. And realistically, as the chain reaction, as PCR progresses, it gets harder to find the primers, gets harder to find the nucleotides, and the polymerase enzyme itself is wearing out too. So there's all sorts of limiting factors that will come into place. Key point, exponential growth does not go on forever, which is good. So if we plot that part of the curve, we have continued exponential growth, and then we hit a plateau as reagents run out. And this basic curve shape, sigmoid curve shape, is common to every real-time PCR reaction, and it's common to every PCR reaction period. PCR is a doubling, the doubling will keep going, and then it will stop, and that makes a sigmoid shape when you draw it out. So once we've seen that, we can relate that to quantities. And here's how we'll think about that using the same thought experiment. What we're going to do is we're going to imagine that you've got a tube of PCR reaction, similar to mine, but your tube, the green tube, you started with 4 times as much DNA as I had in it. and we're going to picture our tubes both at cycle 25 and work backwards. So my tube is the red one, cycle 25, it's got 1 million copies. Your tube is the green one at cycle 25, let's say it's got 4 million copies. You started with 4 times as much. At cycle 25 where we are now, you've still got 4 times as much. If we look backwards though, cycle 24 you had 2 million, cycle 23 you had 1 million. And note there that at cycle 23 you had the same amount of DNA that I've got at cycle 25. The only difference is it took me 2 cycles to make up for that difference. 2 doublings, and I made up for your 4-fold lead on that. A way to look at this is with our graphs, and the original graph I showed was the one of my red PCR reaction, which amplified to 1 million copies at cycle 25. You started with 4 times as much, so for every point on the curve, you've got 4 times as much. What that means is that you've got amplification essentially 2 cycles earlier. Your curve is the same shape as mine, but it comes up 2 cycles earlier because you've got a head start of 2 doublings. We can look at this another way too, that if I've got my red tube, cycle 25 with 1 million copies, if you've now got a tube of PCR reaction with green dye in it, and it has 8 times less DNA in it, then at cycle 25, it's only going to have 1/8 as much. But then cycle 26, it will double, 27, it will double, 28, it will double again. Cycle 28, you've got 1 million copies, you've got the same amount as I had back at cycle 25. So it took you 3 cycles, but you caught up to the same 1 million copy number that I did. And we can plot those curves on a graph as well. My original red PCR reaction, amplified as the red curve. Your tube, the blue PCR reaction, had only 1/8 the amount of DNA, but after 3 cycles, it had caught up to my amount of DNA. So the red and the blue curves, they're identical in shape, they're both sigmoid shapes. The only difference is they're shifted left and right from each other. Yours had less DNA in it, so it took a little while longer to catch up to any individual point on that curve. So what we can see from this is that it's the left-right shift of those curves that is related to the starting quantity of DNA. The more DNA you start with, the earlier those curves are going to appear, and the less DNA you start with, the later they'll appear, because it takes more doublings for those curving curves to cross any particular point. And we can quantify this particular position of the curve by what we call the CT value, or the cycle threshold value. And that's where the curve crosses any individual arbitrary threshold. So in this example here, our threshold is at 1 million copies, of DNA. It's where our PCR reaction finally passes the 1 million copy mark. And one of our reactions, it passed at cycle 23, another one passed at cycle 25, another one took 28 cycles to reach that point. With those CT values, those can tell us the quantity right there, because the quantity is related to 2 to the power of the CT value. Every reaction every cycle is going to be a doubling, so the cycle number just tells us the quantity based on doublings. And like a lot of things in math or biology, we can plot these on a graph. And if we plot our red PCR quantity of 1 and a CT value of 25 on the y-axis, we can plot our green PCR tube that had a quantity of 4. This is a log scale on the x-axis right now, and a CT value of 23. And the blue tube had a quantity of about 1/8 and a CT value of 28. See that all these points, these 3 stars, fall in a straight line. And that's true with real-time PCR reactions. You do a standard curve in this format, and all your standard points are generally going to be on a straight line. For any unknown sample that you have, for example, if we had an unknown that amplified and crossed the threshold at cycle 26, and we could read right off the x-axis what the quantity is. And that's how real-time PCR is used to measure quantities. Just a standard curve and then compare the CTs of your unknowns to your standards. Now, one thing I like to share with my classes is how sensitive real-time PCR is. And keep in mind that real-time PCR is just as sensitive as regular PCR. So ultimately, even a single copy of DNA can be measured. But in reality, it takes about 100 copies to get a reliable signal. So you know you're not just amplifying primer dimers or contaminants or something like that. And an example of what 100 copies would be is 20 attograms of DNA. And not often we use the prefix atto, but that's what it is, 20 attograms. Or if you were to work that out and figure out how much blood that would be in a blood stain, that would be 2/100ths of a microliter of blood would contain about 100 copies of any particular gene. So we all know how small a microliter is. Picture just 1/50th of that, and that's still easily detectable by PCR and real-time PCR. So it's kind of cool to share that with students. They like to see that part. So that's kind of the theory of real-time PCR. Now, how do we actually find out how much DNA is in that tube as it's amplifying? Well, what we use are reagents that fluoresce in the presence of amplified DNA. So we use fluorescent reagents that will only fluoresce when they're near our PCR product. And we're all familiar with one of those products, or one of those reagents, ethidium bromide. It's common and it's well known, but we also know it's toxic. And it doesn't happen to be very bright in terms of fluorescence. So ethidium bromide, what it does is it intercalates into DNA. So it gets in kind of flat ways between the stacks of the DNA bases. When it's in that position, what it does is it becomes fluorescent, so you can shine a UV light on your gel and your bands will fluoresce because the ethidium bromide is bound in there and it's fluorescent like that. That also makes it mutagenic too, because it doesn't come out of the DNA that easily, and it will really interfere with the enzymes that are trying to copy the DNA at that point. Now, an alternative reagent that we could use in real-time PCR, and this happens to be one that's almost always used compared to ethidium bromide anyway, is called CyberGreen-1. It's used because it's got very bright fluorescence. It doesn't take much dye, and it gives a very, very bright signal. The way CyberGreen works is it binds to the minor groove of the double-stranded DNA. So rather than getting inside flat between the bases, it sticks on the outside of the double strands of the DNA. And because of that, that actually makes it much less mutagenic as well. It's hard to see the details in this little graph, but the red bars are the mutagenicity of ethidium bromide. Tiny little green bars on the bottom, that's the mutagenicity of CyberGreen. So CyberGreen is still mutagenic because it interferes with DNA, but much less so than ethidium bromide. The main reason it's used in real-time PCR, though, is because it's got bright fluorescence. So as real-time PCR makes more double-stranded DNA, the CyberGreen fluoresces more. Here's a slide that kind of shows that. As the PCR reaction goes along, and you go from double-stranded to single-stranded DNA and then more double-stranded copies, you have CyberGreen that binds to the double-stranded DNA. And that CyberGreen is illuminated by light of one wavelength. It will fluoresce in another wavelength. So for example, if you hit it with UV light, it will fluoresce a bright green color. And real-time PCR machines can do that. They don't use UV light, they use visible blue-colored light, but they'll still measure the green fluorescence. So how do the instruments do this? Well, real-time instruments consist of three major components. One is a thermal cycler, a PCR machine that does the actual amplification of the DNA by raising and lowering the temperatures. Another component is the optical module, and it will detect the fluorescence in the reaction. And then finally there's a computer, and that computer will collect the data coming from the instrument, and it will also program the instrument with what temperatures it's supposed to reach. One example of a real-time PCR machine is our Bio-Rad Mini Opticon real-time PCR machine. And you can see the two components here. The upper component is the optical tower, and it contains an array of LEDs that can illuminate different wells on a PCR plate and they'll illuminate the reagents in those wells if there's double-stranded DNA and cyber green in that well. It will fluoresce, and there's a detector that will detect the fluorescence. So it can measure the fluorescence in all 48 wells. The lower part of the PCR machine is the actual thermal cycler, and it just raises and lowers the temperatures of the reaction. So it's kind of the workhorse that carries out the actual thermal cycling, or the PCR. And these two components are together in one machine. Another example is the CFX96 real-time PCR machine, and it follows the same basic principle. You've got on the bottom the thermal cycler, does the temperature changes and gives you the PCR reactions. And then the upper part contains the optical module. And in this case, the CFX96, it uses multiple LEDs that can scan over every well of a 96 well plate, and DNA fluorescence is then detected with a set of different photodiodes. So it can measure multiple different colors at once. So researchers can do multiplex real-time PCR where they've got multiple colors in the same tube. So I mentioned the computer. The computer that's connected to a real-time PCR system will be doing a couple of things. The first one is it lets you set up the PCR protocol, so you can program in what the temperatures are that you want each step of the PCR cycle to hit. 95 degrees, 60 degrees, 72 degrees, whatever it happens to be. The next part of the software lets you set up the plate, and the plate setup will tell the software where the unknowns are on the plate, where the standards are if you're doing a standard curve, where no template controls are. So it helps the software analyze the data after. And then the software will also show you what the data looks like as it comes in and then after it's finally collected. So typical data that would come out would be actual traces showing the real-time PCR amplification. If you have standards, you'd get a standard curve. You have a picture of your plate layout, and then a table of numbers that would represent the CT values, that left-right position of the curves. So just a reminder, if you have any questions about the presentation or things that we've covered so far, things that we will cover before the end, just send a text chat to the presenters and we'll try and answer those. So now on to what does real-time PCR data look like and what are melt curves. So this is some actual real-time PCR data. You can see it looks very similar to that theoretical data that I showed you earlier. Each trace has got a nice sigmoid shape. There's amplification. And there's a left-right shift on these curves. The further towards the left the curve is, say for example curves here, they have a lot of DNA. Curves over towards the right have a lot less DNA. And the reason for that is these curves essentially have a head start. They started with more DNA, so they're going to amplify earlier. The final product of real-time PCR data is essentially a table of numbers. The graphs are nice to look at, but it's actually the numbers that let us do the quantitation or the calculations. So for example, for every well that you have, in this case I highlighted well A3, it's going to have the fluorophore cyber in it. It may be a standard, and it crossed the threshold at 8.9, at a CT of 8.9. Another well could be this one, B6, contains cyber as well. It was a standard and it crossed the threshold at 18.69. So without knowing anything else, we could do some quick back of the envelope calculations here. We take 18.69, which is about 18.7, subtract 8.9, the CT value of the other well. That comes up with roughly, say, ballpark 10 CT units between the two wells. Doubling every CT unit, so 2 to the power of 10, that's equal to 1024. So without knowing anything else about these wells, right away with those CT values alone, we can say there's about 1,000-fold difference in concentration between the starting DNA of this one on the top and this one near the bottom. That's really the core of the calculations that come in real-time PCR. is just relating those CT values and the difference between them with a doubling every cycle, just calculating it out. So that's kind of the quantity side of real-time PCR. Another thing real-time PCR can tell us is what. It's not as good as an agarose gel in many cases for telling us what's in the reaction, but it can give us some ideas as to what's in the tube, and that's the concept of melt curves. So melt curves are based on the principle that as you heat double-stranded DNA, it becomes single-stranded. So if you've got DNA binding dyes attached to your double-stranded DNA, as it's heated, those binding dyes will fall off and will no longer be fluorescent. So if we picture our PCR reaction after it's done, we've got a lot of double-stranded DNA. It's cold. The DNA is brightly fluorescent because the cyber green, for example, is bound all over it. If we start heating that PCR reaction up now and taking that DNA product and denaturing it, we're going to have less cyber green able to bind because a lot of the DNA is becoming becoming single-stranded. It's becoming denatured. And ultimately, if we will all be single-stranded and no cyber green will bind at all. So as we heat a PCR reaction that has cyber green in it, what's going to happen is the fluorescence will decrease as we get the reaction hotter. So in this graph here, we've got temperature on the x-axis and the amount of fluorescence on the y-axis. As we increase the amount of temperature in the reaction, the DNA will denature, the cyber green will fall off, and we get a decrease in fluorescence. Now what's neat about this is if the DNA is a different structure in different tubes, for example, you've got a longer PCR product in one tube than another, or maybe a different GC content, then that DNA that's longer or has a higher GC will also have a higher melting temperature. So it's going to take a lot higher temperature before it finally melts. And by taking these melt curves from different tubes, what we can do is see if it's the same DNA that's being produced in different tubes. Commonly, these melt curves will be presented as the derivative of the slope. So instead of the graph on the left, as I showed originally, if you plot the slope versus temperature, you'll end up with a trace that looks like chromatograms. And that's a lot more easy to interpret. And I kind of like to bring up melt curves when I teach PCR because it gets students thinking about the structure of DNA. The GC versus AT base pairing, how GC takes a lot more energy to separate than an AT. Also how a longer DNA fragment is going to take more energy to pull apart than a shorter DNA fragment. So just teaching the concept of melt curves can lead to a whole lot of different discussions that you can bring out about DNA kinetics and things. Melt curves right now have been taken even further with this new field of precision melt analysis, or sometimes called high resolution melt. And in that, a regular melt curve is done with the precision instrument and reagents that bind to every available spot on the DNA. The melt curve is done. It's then normalized. So the starting fluorescence and the ending fluorescence is the same for the melt curve. And then it can be normalized for temperature again in the software. What ultimately happens with this is for different DNA sequences, you'll get a totally different melt curve, totally different shape curve for different sequences. And this can be so specific that it can tell the difference between an A and a T base pair at any given location. So it's commonly used for mutation analysis or mutation screening. Also, it's used in ecology and things like that, where you've got a population of organisms and you want to see how many different species you've got in that population, say a population of bacteria. How many different species are there? You can see right away by how many different curves you have. So that's high resolution melt. So now on to something I enjoy. How do we demonstrate real-time PCR in the classroom? I usually have the real-time lab as the kind of the last section of my short PCR course that I teach. One way to teach real-time PCR is with our Crime Scene Investigator PCR Basics kit. And people who've run that kit before, what They'll know what you do is you do PCR on some supplied DNA samples. You've got different suspect samples and a crime scene sample. You can run the PCR products out on an agarose gel and see the band patterns and match up the suspect with the crime scene sample. So for real-time PCR, you can take that same kit, but instead of using the supermix in the kit, you can use a CyberGreen supermix, one that contains CyberGreen in addition to all the other packed polymerase and nucleotides and things. You can use that CyberGreen supermix and a real-time PCR instrument and computer to run the kit. So you're basically just substituting those components. You can still take the PCR product out at the end and see the band pattern on the agarose gel. But as the PCR reactions are going along, you'll see the amplification happening. And that's really cool for students to see the amplification as it's going on. We've got a application note and a starter kit that are available to help people out with this. And basically it's just running real time as an add-on to the regular kit. Another thing that you can do with the crime scene kit is to do a dilution series with the DNA. Since it's known DNA template that comes in the kit, you can just do a dilution series and see how well the student's pipetting is and see what the amplification looks like, where their curves come out for a dilution series. Because the PCR products in that kit are different sizes, you'll also have different peaks on your melt curve, so you can use that to introduce melt curve and DNA structure and things like that. So this kit is actually great for first-time users because you get DNA supplied with the kit, the primers come with the kit. It's all pretty much known to work right off the bat. Now, more advanced users can do the GMO Investigator kit in real time, and that GMO Investigator kit is one in which you'll do PCR on actual food samples from the grocery store to see whether they're genetically modified or not. There's primers in the kit for the plant DNA and primers for the genetically modified. And with real-time PCR, you can substitute the CyberGreen supermix for the regular supermix, run the reactions on a real-time instrument, and then in addition to the agarose gel that you'd normally run to see kind of a yes-no genetically modified answer, you'd also see the amplification curves, and those positions of the curves will tell you how much DNA was there. Curve comes up early, it tells you there was a lot of DNA recovered from one food sample. If it comes up later, it tells you there was much less DNA in that food sample. We've also got a starter kit available for this and an application note. And this is one I run in my class, and I really like doing this one. The ultimate result of this kit can potentially be a table that you can come up with for different food samples, and what you'll get is the amount of genetically modified food in each food sample. Because real-time PCR goes back to the quantity of DNA, you can calculate all the ratios of different things and get those answers. So this is a good kit for more advanced users. I wouldn't recommend it as a first time because of the big variability in the food samples that you've got. So just to kind of reiterate, teaching real-time PCR in the classroom it actually is really easy. You can use the crime scene kit with the starter package or the GMO Investigator starter kit for real-time. If you're doing your own PCR, say you've been teaching PCR using your own primers, you can just substitute a CyberGreen supermix for your TAC polymerase and run it on a real-time instrument. There's really nothing fancy about the real-time other than you're using a slightly different polymerase. You're also using plastics that the real-time instrument can see through, so you can't use opaque caps and things like that. And you're running the samples on a real-time instrument. So it's really easy to do. You could even design your own primers if you like. To get started with, there's really no difference between conventional PCR primers and real-time. It's only if you're trying to get super optimized reactions that you need to worry about that. So I think real-time PCR in the classroom is cool for quite a few reasons, but here are some of them. One is instant results. You actually see the amplification within minutes of starting the PCR run. You don't have to wait until the next lab period for seeing the results. And anyone who's taught taught conventional PCR knows that you take your samples, you put them into the PCR machine, and then the class ends because you don't have 2 hours to wait to continue on. So it's the next day or the next week that you pick up on that. With real-time PCR, the students get to see the amplification as it's going on. Another thing is I call the what will happen next factor. And that's because the samples are all amplifying at different times. You get students who will want to wait even after class for their own particular sample to amplify so they can see what happens. And that really surprised me the first couple of times I saw it, where students that would normally be the first out the door, they're actually waiting 15, 20 minutes, half an hour after class just so they can see their sample curve come up and amplify. It also does what I call demystifying the black box. Instead of putting the PCR tubes in the PCR machine and closing the door and that's it, now students get to see what's going on there. They see fluorescence increasing in those wells based on what the computer is telling them. And even the temperature control of the PCR machine, the controlling software will show you what temperature the lid's at, what temperature the block's at. And I've seen students get really interested in just that aspect alone. And another thing I've noticed is that it really does connect computers and biology. So students who are into computers, I've had a number of like adult continuing ed students who came from a computer background. They get really excited to see the computer control of the instrument and how the computer collects the data and analyzes the data. So it's kind of my favorite thing to teach, just because it does engage students so much, and it has those kind of real world applicable values. If a student's putting on a resume that they learned PCR, that's one thing. If they learned real-time PCR, that says a lot more. So that's just some reasons why I like real-time PCR. Now, just as a last thing, we'll go over real quick how do we optimize and troubleshoot real-time PCR reactions. And optimization of real-time PCR reactions is important for this reason. Since real-time PCR reactions are based on a doubling of product every cycle, if the reaction isn't optimized, this doubling will not occur. So to get precise calculations of quantity, you need to have a doubling, and to get an exact doubling, that reaction has to be working well. And a well-optimized PCR reaction, when spread out on a standard curve, if you take your template, and will have nice, evenly spaced points on the standard curve, and all the replicates will essentially be lines right on top of each other. So at 100% efficiency, or a perfect doubling every reaction, tenfold serial dilutions will be spaced 3.3 cycles apart from each other. And this graph shows a really nicely optimized reaction. You could get really good quantitative data from that. To optimize PCR reactions, people will normally design multiple primer sets and then empirically test each primer set with a standard curve. They'll dilute out some template DNA, a certain number of tenfold dilutions, for example, and just see which primer set is working best. Then once you've found the best primer set, you can run a temperature gradient to find out the best annealing temperature for those primers. So it's pretty straightforward, and standard curves are ideal for assessing optimization. And this isn't required so much for just demonstrating real-time PCR in the reaction or in the classroom, but if you want to have reactions where you can do quantification, accurate quantification, it's a good idea to optimize, and at least be able to explain to the students how to optimize PCR. So troubleshooting, why would it be important for teachers to be able to solve real-time PCR problems? Well, one is it helps students have better success with their projects, so help reduce frustration. Preventing problems at the start can also help to avoid lost experiment and reagents. And as we all know, being able to explain unusual results leads to great teaching opportunities. So this is kind of another teaching tip that I've noticed. A lot of times students' real-time PCR data isn't perfect, and that gives a great chance to explain to them how experimental design works, how pipetting can be better, how to mix reagents better, things like that. You can teach a lot based on problems that come up as the students are doing their own real-time PCR, if they're doing their own projects, for example. So some basic troubleshooting. A good real-time PCR reaction will have nice flat baselines. The replicates for the same sample will be tightly clustered. The lines will almost be on top of each other. Our dilution series will be spaced out evenly because it's all about that left-right curve position that shows the quantity. So if we make a dilution series, it better be spaced out. The curves should all be nicely S-shaped, because that's the classic shape. Plateau height, I alluded to this earlier, but it really doesn't matter. PCR reactions just double and double, and where they finally end, it can vary over a medium range. So we don't worry about the plateau height. It doesn't have to be the same. The curve should be smooth, and the melt curve should have only one product. So sometimes if things like this go wrong, for example, our replicates aren't tightly clustered, might be pipetting error, or PCR reactions may not be optimized. Samples might be evaporating because they weren't sealed properly. Maybe we've got too much or too little of our unknowns and it's just too little to detect. Or maybe the instrument isn't calibrated properly. Some instruments need calibration. If our baselines aren't flat like they should be, that could also be sample evaporation because the plates weren't sealed properly. Might be bubbles in the PCR reaction that are popping in the first few cycles. Or maybe the reagents weren't properly mixed. Or if the software requires a baseline window to be set, maybe that wasn't set properly. Our dilution series may be messed up. If there's pipetting errors, we may have the curves coming too close together or too far apart. There may be too much DNA, there may be PCR inhibitors, maybe even too little DNA, or the PCR reaction just isn't that efficient. And if the curves aren't S-shaped, sometimes curves can have really strange shapes. Maybe it's not actual PCR products at all. Maybe PCR didn't even work and it's just a sample that's evaporating that you're seeing, causing the fluorescence to change. Or it could be fluorescence drift in unamplified samples. The cyber green may be bleaching out. But in any event, there's something seriously wrong with the assay if you don't see those sigmoid curves. And if the curves aren't smooth, again, it could be poor pipetting or evaporation. Maybe the assay's got problems, or maybe even there's like electrical noise on the causing problems with the instrument. Melt curves, if we have multiple peaks in our melt curves, that tells us that we're amplifying more than one product. Might be normal if we're making primer dimers at very low concentrations. But maybe it tells us that we're using the wrong temperature for our primers and they're binding at a different site and making extra product. It is a suggestion that primers need to be redesigned though if we start seeing multiple peaks in our melt curve. So common themes that crop up here: care in pipetting, be careful in choosing the plastics and sealing the plates. Care needs to be taken in experimental design, and proper use of positive and negative controls. So this kind of goes into another teaching tip. You can use real-time PCR to teach the importance of properly designed experiments. It's a great learning experience to go through looking at real-time PCR data and not knowing what's going on with it because there weren't positive and negative controls. It really kind of reinforces that you need to have things like that in there. And for any of these troubleshooting things, our technical support group can help you. So you're definitely not alone in resolving these issues. So we've looked at these subjects today. What is real-time PCR used for? How does it work? What chemicals and instruments are used to detect DNA? What does the real-time data look like? How can we demonstrate it in the classroom? And what are some ways to troubleshoot real-time PCR? And with that, I'll turn it over to Ingrid for a moment.
Thank you, Dave. Before we answer questions, I'd like to let you know that there are several resources available to you regarding real-time PCR. These will be sent along to you with the playback of the webinar in a few days. Also, we'd like to extend a special offer to each of you. When you purchase the real-time instrument of your choice, you can receive a free real-time PCR starter kit also of your choice. That's over a $500 value. please contact us at biotechnology_explorer@bio-rad.com to receive your quote for this special offer. Also, at the end of this session, you will have the opportunity to complete the exit survey to give us feedback on this session and to share other topics you'd like to have us present. And now back to you, Dave, for questions.
Thanks, Ingrid. Okay, so a bunch of questions have come in. One good one, is the animation of PCR available for download on the Bio-Rad site? I'm pretty sure we do have that available, right? Yeah. So we can put the link to that in the email that we send out after this. Another question that came in is this the iCycler software that was shown in here? That software, the screenshots I had were actually our CFX Manager software. That's our newest one. The iCycler software is almost identical in terms of the workflow. You still have to set up the protocol on the plate, and you still get the same looking result. Really, any manufacturer's real-time PCR is going to have that same basic workflow to it. Another question we had is there a standard threshold or is it chosen? So in that thought experiment I did, the threshold was 1 million copies. Everything was based over when those curves passed the 1 million point. And what the software will generally do is it will pick a threshold point where all your curves are essentially parallel to each other. So the software will typically choose the threshold. You can always choose your threshold manually, and basically it's either by using a standard curve or by just looking at the data by eye and looking at where your curves are parallel to each other. Let's see. Oh, this is a good question. Someone asked that they're not clear on how a CT value can be anything other than a whole number, because PCR is a set of discrete whole cycles, and that is true. The PCR cycles are discrete, but what happens is the amplification curve, being a logarithmic curve, it could cross anywhere between individual cycles, and it actually does. The curves are smooth curves. So when you connect the points on the curve, those points will fall just anywhere between those cycle numbers. And you can actually tell the difference between, typically around 0.1 CT units. So a tenth of a CT or a tenth of a cycle, you can reliably tell the difference for a well-optimized assay. So it doesn't have to be whole numbers, but tenths of numbers is about the limit for the detection or the sensitivity on telling one sample from another. Oh, there's a question about the practical advantages of real-time PCR over conventional PCR. And it depends on what the goal of the experiment is, or the goal of the research. So, for example, the container shipped full of GMO grain, the only way you could tell the percentage of GMO in that using PCR would be to use real-time PCR because of its quantitative nature. If you used conventional PCR, you'd certainly have a good chance of detecting genetically modified food in there, but you'd never know if it's above the 5% legal threshold or not. The same with, say, forensics. If you were looking for just did a blood stain contain a DNA type that matched a suspect, you could do that with conventional PCR. But if someone was to turn around and ask, well, how much DNA was there? Was there really enough to accurately detect? To do that, one of the ways to do that would be real-time PCR. So conventional PCR, useful for anything where you want to tell what the sample is or what the template was, but real-time PCR, it's needed for quantifying it or telling how much there was. Oh, there's a question that came in about how many students would one typically have for the GMO experiment. And when I run that kit in real time in my class, I would typically have around 15 or so students, I'd split them up into pairs. So we'd have maybe 7 or 8 work groups, something like that. If you end up with too many students, and this is true for any teaching lab, too many students, and you can't really give that much attention to the individual students that may not be following the whole thing. So I'd say for that, because of its complexity, best to keep it under 10 work groups if you can. Just looking at some other questions here. Oh, okay. Someone asked, do we have a recommended site for purchasing primers? Right now, Bio-Rad doesn't sell or make primers, other than the ones that we supply in our kits already. So places that we recommend people to get primers from would be something like IDT Technologies or also BioSearch Technologies. Both of those companies do a really good job at developing and selling primers. Operon also has primers too. Someone asked, can we make a synthetic DNA from RT-PCR? There's actually 2 RT-PCRs. I'm not sure which one that refers to. One is real-time PCR, and the other is reverse transcriptase PCR coming from a messenger RNA sample. Either way, the end product is DNA. So no matter what you start off with, you're going to end with DNA. And that DNA can be put on an agarose gel. It can be used in anything you'd use DNA from a conventional PCR reaction for. Someone asked, is that the maximum curvature approach? The maximum curvature approach, that's the kind of technical terminology that's used to identify how a threshold is set. So that bar that I had at the 1 million amplicon point, when that's automatically set in some software like our iCycler software, that's set on the maximum curve of the maximum point of curvature on most of the traces. So essentially it's a fancy way of saying where the curves are all amplifying the most, where that exponential increases the most, that's the best place to put the threshold. Had a question, can we record this presentation? The presentation has been being recorded already, and it will be available for download at some point after. We'll send out the links after. Oh, yeah, I like this question. How much background do the students need? From someone who currently teaches 9th graders. I have actually taught in one of my PCR classes continuing ed students, one of which had very little background, definitely no college background, and I don't think, if I remember right, he had finished high school. By the end of it, he was actually very comfortable with how DNA structure was, how regular PCR worked, and how real-time PCR worked. So even though a class may not understand all the concepts or all the levels, I think a lot of these things are applicable to a wide range. And because real-time PCR touches on so many different things, the structure of DNA, how to draw graphs, how to analyze graphs, how to design experiments, how chemistry works, I think it can be used in a lot of different situations. Okay, looks like we just have time for 2 more questions now. So one question came in, are all of the machines capable of precision melt analysis, or do you need a special thermocycler? And the answer to that is that it does take a specialized thermocycler for precision melt. Regular melt analysis can tell the difference between, I don't know, dozens of base pairs, but if you're trying to tell the difference in a single base pair, you need a cycler that's got a very precise block on it, very precise temperature control, and that can also be calibrated for any differences from one machine to the other. And our CFX96 system, it can do precision melt analysis, but our other systems like our Mini Opticon or our iCycler series, those ones you wouldn't be able to do the most precision of the precision melt analysis on. And let's see, we had a question about the bubbles in the wells. How much of a real issue is this? It varies. I help people with real-time PCR every day when they call in through tech support. I'd say every 2 or 3 days I'll encounter someone who sends in data where the problem was caused by bubbles. The easiest way to avoid that, and you can use this for lots of things in your lab, is if you do what's called the reverse pipetting, where you pipette down to the second stop on your pipetter to suck up the reagent, and then pipette down to the first stop to expel it into the tube. And that way you're not expelling a bubble in. But anyone who wants more details on that, they can just contact me after the fact, and I can help them with that. So I think that's all the time we have questions for. All the other questions we will answer and we'll put into the email that goes out after this. And thanks everyone for attending. It was a lot of fun talking to you.